Welcome to my presentation, Unlocking the Banana Pangenome, Harnessing Genetic Diversity. Worldwide consumption of banana is predominantly from the Cavendish subgroup, which is a triploid cultivar and therefore unable to reproduce sexually. Instead, this cultivar is being propagated via identical clones. Therefore, the genetic diversity within the Cavendish subgroup is very low. KeyGene is partner in the Yelloway initiative launched by Chiquita that aims to breed for new banana varieties that exploit the genetic diversity present in nature. The two most important fungal diseases threatening worldwide banana production are Fusarium Tropical Race 4, also known as the Panama disease, and Black Sigatoka. These diseases cause huge losses in the banana industry and require spraying large amounts of fungicides. Within the Yelloway initiative, we are breeding new banana varieties that are resistant against these diseases. To do this, we exploit the genetic diversity present within banana. We sequenced 150 mainly diploid accessions and also constructed a high-quality reference genome of Cavendish. By using admixture analysis, we could reconstruct the Cavendish genome and select lines for it in our breeding program. Using these lines, we make crosses in our greenhouses between resistant and susceptible accessions and as such can generate mapping populations. The progeny is tested phenotypically as well as genomically by using adaptive sequencing from Oxford Nanopore technologies. And as a regions of interest, we select resistance gene loci. We achieved 5 to 6-fold enrichments on the target regions involved in disease resistance, and this allows screening large germplasm collections very cost effectively. Here you can see an example of 2 banana varieties, and we are able to really phase the both haplotypes using this adaptive sequencing approach. The main results I will show here is on the construction of a banana pangenome. There's 2 aims in this project. The first one is to generate chromosome scale telomere to telomere phased reference genomes of 9 mainly diploid accessions and 1 triploid accession. In addition, we generated draft reference genomes for another 50 accessions. We used Oxford Nanopore technologies for the data generation. We generated both simplex as well as high duplex data, and for 2 accessions we also generated long-range multi-contact Pore-C data. For the data analysis, we used de novo assembly and variant detection, we used the KeyGene pangenome tools, and for visualization we used KeyGene CropPedia. Here I will focus on the chromosome scale phased reference genomes. So we selected 10 banana accessions from 5 subgroups. For the data, for 1 accession we generated hi-fi data and nanopore duplex ultra-long data in combination with Pore-C data. For all the other lines we generated nanopore duplex data and nanopore simplex data that was filtered to 50 kb and longer and actually was generated from the same duplex flow cells. We used reference-based scaffolding for these lines. So here on the right you see a data overview of 8 from these 10 lines. So for 1 line we used hi-fi data, and what already clearly strikes is that the read and 50 of the hi-fi data is almost half of the unfiltered duplex data for all of the other lines. The tools we used for analysis were hi-fiism for de novo assembly in combination with raft, RegTech for chromosome scale scaffolding, for compleasm for determining the gene completeness, and Mercury for determining the consensus accuracy using 25x of Illumina data. Here I show a result of one of the lines, KeyGene 002, of which we used hi-fi data in combination with nanopore duplex data. So we used hi-fiism and draft. So initially we generated the contig level assembly, which was already of very high quality, having around 100 contigs for each of the 2 haplotypes. And they aligned very nicely to the publicly available telomere to telomere DH per hang line. Once we added the Pore-C data, we already could achieve chromosome scale assembly for both haplotypes, which is also shown here by the statistics. So we could determine about on half of the chromosomes telomeres ends, and also the consensus accuracy of these assemblies was very high. Here on the right you see a Pore-C interaction map for this haplotype phase assembly. So the longest duplex read we could map with a very high accuracy to this genome mapped to chromosome 5 on haplotype 1. And mind you, this read length is about 8 times the length of the Hi-Fi read length. So we used Siri to zoom in on the larger translocations we could identify, actually on both haplotypes. So on haplotype 1 we could identify a large translocation going from chromosome 1 to chromosome 4, and on haplotype 2 there was another translocation going from chromosome 1 to chromosome 7. So nicely, these translocations were already described in a publication in the Plant Journal in 2020, nicely confirming our results. Moving to the second genome I show the results of. This genome we assembled using only Oxford Nanopore data in combination with HiFiasm and Raft. So we generated 26 gigabases of Nanopore duplex data and 15 gigabases of 50 kb simplex data derived from the same flow cells. And here we use RegTech for reference-based scaffolding. So also here, the contact level assembly was very contiguous, but once we used RegTech for reference-based scaffolding, we could again achieve chromosome scale references for both haplotypes, and actually for one of the haplotypes we were able to identify all the telomere ends. Also here the consensus accuracy was very high, and this was even without using polishing tools like deep variant. So also on this genome assembly we could identify one very large translocation. In this case it went from chromosome 3 to chromosome 8 compared to the DH telomere to telomere Bahang genome. Also this translocation was previously described by Martin et al. KeyGene has developed its own toolset for rapid pangenome construction. It consists out of multiple steps. As a first trial, we used as input our 49 draft reference genomes generated with simplex data, and we also included some public lines. So we performed the progressive multiple genome alignment, the pangenome construction, the extraction of the genetic variants, and we visualized the results in our CropPedia package. So as an example, we zoomed in on the resistance gene analog 2, and actually there we could nicely confirm the insertion of repeats in the promoter region of this resistance gene, which actually has been described end of last year in a very nice publication in Nature Genetics. But as opposed to just a few lines, in this case we used 49 lines. So this provides real insight, new genome insights. Finally, here I show a series plot of the malacensis subgroup, in which 3 of our fully phased reference genomes are depicted, and on the top line the public Bahang version 4 reference genome is depicted. Here I show chromosome 3 and chromosome 4. What you clearly can see is that on the edges the synteny is quite high between these chromosomes and genomes, but moving more towards the centromere you see larger structural variations occurring. These are initial results. Currently we are finalizing the analysis and expect to use these results in our breeding efforts. This was the end of my presentation. We believe the future of banana is diversity. Thank you for your attention.